Hello, welcome to our second lecture of our lecture series on single-cell high-throughput data science. So in today's lecture, we are going to discuss how can you get UMI count matrix out of this single-cell RNA-seq data analysis. That's gene expression information from individual cells. We will mention how can we do the QC, quality control of the raw data. At the end of the lecture, my student, Daniel Osoro, will have a live demonstration of using the single-cell pipeline to from a 10x genomics called the Cell Ranger to do this on the computer, and I'll see how exactly that pipeline will work. So as I mentioned last time, and we have a exponential growth in our capacity of sequence number of individual cells. So this exponential increase end up in the 10x genomics commercialization of the droplet sequence technology in 2013. So this is a dominant platform we are going to use. So I will focus on introducing this technology. As we mentioned, this based on the droplet technology, using partition OU to separate individual cells and load onto this Chromium controller to prepare your library. So the first problem we will encounter in your real data, in your real single-cell experiment is single-cell preparation. There are technical considerations. How are you going to dissociate your cell and make them separable and captured by the partition oil droplet? For different tissues, you may want to consult in different protocols. So their specificity about the concentration of your cells, number of cells, your preparation. Then you have to calculate the percentage of recovery. And if you have aimed numbers of cells, for example, you aim to sequence at least 5,000 cells, you may want to aim a little bit higher, say 7,000. Then after your experiment, you eventually get 5,000 individual cells information. So there are technical considerations, but you have a lot of reference, and the protocols can look at. Some tissues will be more difficult to dissociate, but it really depends on which tissue, which cell type you are working on. So once you are ready, and there's a mixture of the sample with master enzyme mixture, then you will have to load them into this 10x genomics Chrome preparation, this panel. And another component will be the GLBs, will be loaded here, and then partitioning OU will be loaded here. And then you put this back to the Chrome controller. It will prepare the sample for you and give you the prepared library for each sample in this mouse here. Okay, the whole process maybe only take 1 or 2 hours for preparation, and the rest of them will be sequencing your library using next generation sequencer. So I'm going to explain the principle of this preparation. How are you going to prepare this GLB and prepare your cell and enzyme to let the barcoded GLBs to be captured by the partition OU and to prepare those OU droplets. It's called the GEM. How the GEM is structured, how the barcode to let you to separate each individual cells, and also each individual gene's expression. So GEM stands for GeoBead Emulsion Okay, so this is the structure of this GEM. In the center of this GEM is a gel bin. It's a reagent provided by the 10x Genomics. And also, you will have enzyme in the cell. That's your sample. Hopefully, most of the GEM capture single cell in there. Sometimes you have double cells, but from analysis, you can separate them out and get rid of them because you want to use the single cell information. Okay, these GeoBs are covered by a lot of DNA fragments, full of the DNA fragments. So if you look at the DNA fragment, it contains this primer sequence, and then next is called the 10x barcode. So this 10x barcode will tell you which GeoBid it is covered. So all the DNA fragment of this fragment contain the same barcode. So for this particular GeoBin, the 10x Genomics barcode will be the same. There are 16 base pair. Then for each barcode, then the next fragment is a 10 or 12 base pair UMI barcode. UMI barcode will be unique for every this fragment. Okay, so this is ligate next to the 10x Genomics barcode. And after that, there's the poly T tail. This will be used to fish out, to hybridize to poly A tail of the mRNA generated from the cell. Okay, so this is the structure of the GEM for the one GeoBin. So for your sample, each GeoBin captured one cell around itself, captured by the oil droplet. So after the cell has been lysed, all the poly A-tailed mRNA have been released. Then, because you have a poly T tail here, so those poly A tail will be hybridized, and then the PCR reaction will happen to amplify this whole fragment. Okay, so UMI stands for unique molecular identifier. So each mRNA molecule will be identified by this unique orange barcode called UMI. So the 10x cell barcode has 16 base pair. That's this green part, this barcode. This is unique for each of these GeoBs. And then they have a standard sequence for this cell level barcode. So this is the barcode sequence. So you can see this from this file I download, and that's the top. It's not from AACT, and this is unique until this. So in theory, you have a capacity to separate such number of cells individually. In principle, if you have a one GeoBid capture one cell. Then another system is the UMI. UMI is 10 base pair long in V2 version of the solution of the chromium single cell 3' solution. In version 3 of the chemical, they increase the length by 2, so you have a 12 base pair. So that's from the real data. We can see each gene can be identified by this 12 base pair. Okay, great. So once this PCR reaction happened in each individual droplet, the sample will be put together. Then the next step will be a bulk library preparation. So this is very similar to traditional single-cell RNA-seq. The only difference is your template is not mRNA or reverse translate the cDNA itself. It contains this cell level barcode and also UMI. So you add the enzyme, add these fragments, raise 1, raise 2, and make the library ready for next generation sequence. You can, after preparation, this is called a post-GEM library construction. After you finish this step, you can sequence your library using any favorable Illumina sequencer, illustrated as here. So this will give you some details. What are we going to sequence? We will generate 3 arrays, 3 files. So array 1 will contain UMI and also cell barcode. Array 2 will sequence the mRNA itself. Array 3 is called index, is a sample barcode. So that's the illustration cartoon, and as you remember, the cell level barcode, 10 genomic barcode, is 16 base pair, and the UMI is 10 or 12 base pair, depends on which version of the chemical you're going to use. So the array 1 will be either 26 or 28 base pair long. So this is a very short fragment. The rest of them will be cut off by the downstream software analysis. You only need the information of UMI and the cell barcode. Cell barcode will tell you which cell the rest of the sequence come from, and UMI will tell you which gene, whether there's a PCR replicates or not. If you have the same UMI, you will merge them into one because there has to be unique. And race 2 will tell you where which gene the mRNA comes from. So you compare your race 2 to the transcriptome of your species, and then you will decide which gene you are reading from. So that's the same information I just talked about, but it just uses slightly different illustration. You read 1 is here, 28 base pair if you use the bursary version, and the rest of them will be top poly A, will be get rid of in the analysis. And your race 2 is here, is on the genome, on the gene mRNA itself, transcript gene. It may contain 90 base pair. That would be most of the time will be enough to let you know which gene this cDNA was translated from, transcribed from. Then you have this index, sample index, which is not very important. So here I want to use this example to show you why UMI is important to remove the PCR duplicates. So in this particular example, we have a UMI in bold, and then we have a cDNA sequence, which is sequence from a particular gene. They're all the same, so we know this is from a gene X. Now my question is, how many counts are there for this gene X? In other way, I want to ask the number of UMIs. UMIs for this gene, for this example. The number of UMI will be proportional to the expression level, gene expression level. Okay, so this number will be used in our matrix to tell for this particular cell, the gene expression level of gene X is whatever this number is. So how are you going to count? If you count this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, looks like there are 10 transcripts have been sequenced, but this is not the real 10 is not the real gene expression level for this gene. The reason is we need to consider the PCR amplification bias. So if you look at more carefully, these two have the same UMI. So we need to count this as 1. And these two have a same UMI. It's another 1. And this is unique. So we will count that as 1. This is 1 unique. This is unique as 1. This is unique 1. And look at these two. They are identical to these two. So we already counted these ones, so we will not count this at all. Okay, not this. So at the end, we have 1, 2, 3, 4, 5, 6. So the UMI for this number of UMI for this will be 6. Okay, so this gave you some idea, use real example to show you how this will be removed, how to remove this PCR duplicates. Another thing you want to consider is the sequence dips. We recommend you sequence per cell, you sequence 50K, that's 50,000 of row rates. So this is a saturation test. If you sequence up to 50,000 rates per cell, then you can for this particular cell type, you can identify more than 3,000 genes on average, medium. And for this particular cell, you may identify nearly 5,000 genes. This is the same kind of analysis, but using 2 different other cell types. So if you sequence up to 50,000 rates per cell, then you will have over 1,000 genes that have been detected. And for this one, you have 2,000. So this sequence dips is cell type dependent. If you sequence enough, some of this will reach to the saturation. You don't see the increased number of the gene, detect gene a lot once you sequence up to, say, 150,000 rates per cell. But anyway, the sequence dips is something you want to consider when you design the experiment. See, once you have got your sequence, then say from the controller, and then the center software we are going to use, provided by the 10x Genomics, is a cell called the Cell Ranger. They also provide the software support for the controller itself and also the post-processing package looper browser. But they are not essential. We don't need to care about this, and the center will be using this to do the mapping and to get the rays, the expression matrix out of it. So that's a pipeline, and unfortunately, it only runs on Linux. So if you really want to use it yourself, you have to have a Linux machine ready. And it's a little bit computationally intensive, so you don't expect to get the results in a couple minutes. You probably have to run your sample overnight before you can get your expression matrix. So that's the end product of this pipeline, is a gene cell matrix. So that will be a matrix of genes and the cells. Each element of this matrix is gene expression of this gene in this cell. So the Cell Ranger will read the UMIs, the 10x Genomics barcode to process those barcodes, and then it will use called star aligner to map your rays to the gene. So to get the transcripts, the number of transcripts, and then compute the UMI. At the end, you will get this. It also can be used to do the sample aggregation. But the main pipeline is doing just this. Okay. So the input for Cell Ranger is 3 FASTQ files generated from your Illumina sequence. So you see this part is all the same. To tell you that from the Illumina sequence, tell you the sample name, the length, and maybe sample names here, index. Then you have an index. So this index file, the first one. And then you have R1 file. Okay, R1 file will be small because it's only have a 26 or 28 base pair per rate. This is a barcode rates. It's all here. And R2 contains the information from mRNA. It's in here. So that's 3 input files a Cell Ranger will ask for. Then you have a Cell Ranger count, command line, and Cell Ranger aggregate if you want to merge multiple runs. But I will leave those details to Daniel, and Daniel will demonstrate how I'm going to use Cell Ranger. He's going to use the download examples, and on the Linux machine, I will demonstrate how exactly you are going to code and to get this information. At the end, Cell Ranger will generate a report. Okay, so the most important information will be here is how many cells, estimated number of cells, has been generated from the running of this 10x Genomics. Cell Ranger will get, in this particular example, you generate the cell for 15,000 of them. And then another important information is average genes per cell detected. Okay, so this you detect the medium number of genes detected, over 2,000 of them, and you have mean rates per cell. So this gives you the information of the diffs. And there are other informations about sequencing, about mapping, and your sample. They all are provided in this report. Okay, so as I said, next we are going to show you how to use Cell Ranger in Linux. And Daniel will be demonstrating these pipelines, and the Chen, both of them are my graduate students, will help Daniel to show you how to do it. All right, enjoy.
Okay, yes, it's recording. So, okay, so today I'm going to show you how to quantify the expression of each gene in each cell using the tool provided by 10x Genomics. The tool can be downloaded from resources, and you can go to datasets, and then you have the single cell, and okay, this is the data. Let me see. Well, it is easy if we just Google Cell Ranger.
Okay.
And this is the package that we are going to use. Today we are going to use two of the tools included in Cell Ranger. The first one is make a reference, because we are going to personalize our reference to include a transgene, and also the Cell Ranger count. The FASTQ files that we are going to analyze today were already processed by the core facilities, so they already run this and provide us the FASTQ files.
Okay.
So what we are going to do is to install it. To install it, we go to the single cell gene expression software, and we have a download page from here. They provide the direct link for the Cell Ranger. So we need this command line. So we copy this and we paste in here. It's not working.
So TX doesn't have Cell Ranger installed?
By default, no. I have it in my own.
Okay.
What is that you're using?
The supercomputer that the department provides to us.
Oh, okay.
So I just need to copy this link here, and it will start downloading the Cell Ranger. And I will do the same. This sample is for mice, so I will download this other reference that is designed for mouse musculus. This
is a FASTQ file?
This is the reference for a star. So it includes the FASTA file for the genome, the GTF file, and they also do the pre-building of the library. So it's like a pre-compute reference for Cell Ranger.
I see.
Okay, so I download the file, is this one, Cell Ranger, and I also download the reference data for the mouse musculus. I place the FASTQ files in this folder FASTQ, and I downloaded the file for the TD tomato sequence, that is the transgene I'm going to add into the reference, and the GTF file. These both, I put them available for you in my GitHub. So they are here in utilities. I made a tag sequences folder, and I put all the possible transgenes that usually people use in the lab.
Okay.
I put the TD tomato FASTA file here, see?
Okay.
And I also compute the GTF file. So I put the len, the the line where the gene is located, the names, everything is ready for you. You just do row, copy this link, and in here in the terminal you just type curl minus capital O, and you paste the link, and it will automatically download the file.
Okay.
Okay, so the next step is we are going to uncompress the reference data because we are going to personalize it to our own purpose, that is adding the TD tomato gene.
Okay.
So we are taking the reference that Cell Ranger provides, and we are going to personalize it to create a new one.
So that reference is the genome FASTA file and GTF?
Yes, but it's the genome without the TD tomato gene.
Yeah, so the TD tomato you're going to add is another contig or another chromosome?
I'm going to add another chromosome, yes.
Okay, I see.
So I am going to uncompress this.
So it's just like command lines. Is this what I'm going to learn like next semester in that class?
Yes.
Okay.
Yes, like command line techniques are this, like how to move between folders without seeing it, just typing.
Okay.
Okay, it will take a while because the file is 9 gigabases. But we don't need this portion anymore, so I'm going to do this full screen.
So this reference, we decompress it and it will be ready to use for STAR?
For Cell Ranger, yes, for a start. But the point is, it doesn't contain the TD tomato. So you can use it if you don't have a transgene in your sample, but in this case, the professor wants to quantify also the TD tomato.
Usually when I want to add something to the reference, I will do that before index. Before build like STAR generate genome before this step.
Let me show you how I do that. Well, I use this reference because it's easy for me to get all the files and all the stuff. I just take what they provide to us and copy that in here.
And move it to the text folder.
Yes.
I mean, so you can add another chromosome after you build index or build the genome using that, right?
No, I'm going to take the data they provide because they already have like everything sorted and ready to use.
Yes.
Just going to concatenate this genome.fa with this tdtomato.fa.
Yes.
And tag genome.
Yes.
And I'm also going to take the GTF file that should appear next here with this GTF.
Okay.
And generate a tag GTF file.
Yes.
And then I'm going to make the reference again.
Oh, I see. How do you add these two together?
I will show you. Just give me a second, it can uncompress.
Okay.
We are being recorded, remember? It's fine. We can cut this portion.
Oh, can you edit?
Can I add? Can you
edit? Can you edit the video?
Yes, I can edit the video.
Okay, I see.
I can cut portions in, at least in my iPad, I can. So I will delete all these previous steps. So since we say like we are going to uncompress the file until it finishes, I will remove that.
I see.
Okay, so I'm going to concatenate this GTF file, genes GTF, with this TD tomato GTF, okay?
What is concatenate?
Okay, let me show you what is concatenate. I have
Okay.
a file in the FASTA file called genome.fa.
Yes.
So what I'm going to use is just show you the base, the last line in that folder, in that file. So I will use tail, and this is how it ends.
Yes.
Now I'm going to concatenate both of them. And then I'm going to use cat, that is the concatenator. I'm going to put the first file, that will be the genome, and then I'm going to put TD tomato FASTA file. And with both of them, I'm going to save a new file that I'm going to call tag genome.fa. Okay?
Okay.
And I will do that.
Okay.
It won't take too much time. It's done. Let me show you the last line of the tag genome. Now is the TD tomato. You see?
I see.
Okay, so it is already concatenated. And I'm going to do the same for the GTF file. So I'm going to concatenate this genes GTF with the TD tomato GTF. And I'm going to make tag genes.gtf, okay?
Okay.
And that's all what I need from the reference provided by Tenet Genomics. So I'm going to delete those two folders in order to save memory. So remove the reference. And these are the two files that we generate, tag genes and tag genome.
Okay.
Okay?
Mm.
Now I'm going to uncompress cell ranger.
Next one.
It takes more time than what I was planning to. The worst thing is I have everything out of the folder already.
Oh, you're just gonna
Yeah, I'm just doing this for showing the sample. And I could.
So you already have a cell ranger installed?
Yes, I do have everything done.
So let's we can use that one.
No, just wait.
Okay.
I need to show every step
Oh, okay.
because you may forget what happened here. So
Okay.
it's done. Okay. Now we have the cell ranger. I'm going to delete the compressed file to save space. So inside the cell ranger, there is a binary folder here. And inside that folder, there is cell ranger. Okay?
Okay.
So what I'm going to do is to add that address to my path so I can type cell ranger and access to the package without needing to go to the folder. So to do that, I'm going to use a export path equal to the position where I'm located. I can access that position with PWD. So I'm in this place. I'm going to do export path equal to data DCO speed cell ranger and then bin. And this will be added to the path. So I'm going to do this, and now I can type cell ranger, and it will be here. So
okay.
What I'm going to use is this tool to make the reference.
Okay.
So it's cell ranger make ref, and if you type it down, it will let you know which ones are the arguments that are required to provide. So it's the name of the genome that you are going to create, the FASTA and the genes. So I'm going to do this. Make ref minus genome, and I'm going to use TD tomato musculus 10. Okay? I decide what is the name. You can decide your own. Minus FASTA, and then I'm going to provide the file FASTA that we create. Tag genome FASTA, and then I'm going to add the genes minus genes, and that will be tag genes.gtf. Okay. So if I do this, it will create in the new reference TD tomato mm10. It will create a new folder. And this is what it is doing.
How long does this take?
Like 5 minutes.
Oh, that fast?
Yeah.
But in STAR, when you build a genome, it will take like 30 minutes.
Okay, let's wait.
Okay.
If not, we can cheat.
Huh?
If not, we can cheat. We can copy the folder I already produced and make it work.
Okay.
Piece of shit. Being recorded, sorry.
Yes, we can cheat. Copy the folder that I already produced to here. And this reference is the one that we are going to use to quantify the genes in each one of the cells. Okay, there is an error in here. It says like invalid numbers. And I'm going to modify that GTF file. So nano td tomato.gtf. Oh, I use. Okay, I'm going to do this. I'm going to copy my reference from the other folder and copy to here. So rm minus td tomato mm10. Then copy tag mm10 to td tomato to here.
So this one is the pre-built reference you
is the one that I made before. Yes, I guess in GitHub the tabs are count like 2 times the space, and it needs to be a tab in Linux. So I can show you how troubleshoot that problem.
Okay.
Is you just open the file and you use tabs to separate everything.
Okay. So the GitHub file you generate it in Windows?
Yes.
Oh, and it cannot it is not compatible with Linux.
No. Like everything, every time that you have to compute something in Linux, you should do it on Linux, because if you do it on Windows, the way they process the spaces and the new lines are different.
Yeah. Okay.
That is like the main problem I'm having when I work in both systems at the same time.
Okay.
Okay, just wait a second. Okay, so now I'm going to move this tag and10 to td tomato and 10, and we are good to go. Okay, so now what I'm going to do is to use cell ranger count to quantify the expression level of the genes. So if we take a look of the FASTQ files, they all have a pattern in the name. So it's YG2S2, and then the line and the index, the read 1 and the read 2. This was sequenced in 4 lines. So there is a line 1, 2, 3, and 4. And each one has their own index, reads, and stuff. So this portion of the name of the file is what we need. So I'm going to use cell ranger count minus ID, the transcriptome minus the transcriptome. The transcriptome I'm going to use is this td tomato mm10, and the FASTQ files that I'm going to use are in the FASTQ files folder. So if I do this, FASTQs, if I do this, it will run out and it will generate the counts for each gene in each one of the cells. And that's all. That's all what you need to do.
And the output will be a cell expression matrix, like gene cells?
Let me stop here, and then it will run for 4 or 6 hours, and at the end it will return you something like this.
Like a BAM file?
No, yes, it will give you a BAM file, but okay, I have some examples in Windows. Here. It will return a folder for each one of the samples. In this case, it will be just one. And then inside that, it will be these folders. So the web summary contains all the information about the sequencing of the sample. It will give you the number of cells, the number of reads per cell, the median, and all the statistics about the sample. Okay?
Okay, but when I remember when you pay for the cell ranger, it will automatically gives you the HTML file.
Yes,
so,
this HTML file with the analysis in here. You click here and it will give you the tSNE projections and all the clusters, the cells, and all the stuff.
I mean, will they do this cell ranger count for users?
No, they just give you the FASTQ files. And what you inside this folder, there is one folder called filter feature barcode matrix. So this contains the barcodes, the features, and the matrix. The 3 files.
I see. So the matrix.mtx is the gene by cell matrix?
Yes. And the features are the genes, and
yes.
the barcodes are the IDs for the cells.
I see. Okay.
Okay, that's the result. So you will need to provide this folder for each one of the samples that they give to you.
Okay.
Because it contains the BAM files and every intermediate step is in here.
Oh, I see. Okay.
Okay?
Okay.
Yeah, and that's all. All the information that is in this file is also in this Excel spreadsheet. So you have to concatenate several samples. You just need to copy and paste all the rows in here, and that will give you all the information that they will need. Okay? All these statistics are in this Excel file. And that's it. Do you have any questions?
No questions.
Okay. So let me stop recording.